Our next speaker is Shawn Kaeppler from the Department of Agronomy at the University of Wisconsin-Madison and also the Great Lakes Bioenergy Center. Shawn has been on the pointy end of the stick addressing some of the issues that Bob Goldberg outlined this morning, understanding how we can more rapidly get more out of the plants we need, both for food and, in this case, for biofuels. So today, Shawn's going to talk to us about genomic analysis of variation in seed and plant size in maize.
Thank you. Thank you for the opportunity to talk here. I'm going to give you a little bit of information on some of the technical observations and some trends we're seeing in a higher eukaryote, one of the more challenging genomes. I'll try and acknowledge the people as I go along, but I wanted to, at the beginning, acknowledge some of my key collaborators, Natalia DeLeon, Robin Buell, and Dan and Ufi here, and especially Carrie Berry for helping to organize all our projects. We're working on maize and switchgrass in the Great Lakes Bioenergy Research Center. Switchgrass, of course, is a more recently domesticated crop species. Lots of complexity, so we're using maize and some other grasses as models. I'm going to talk today about some of our results from maize. In our studies of natural variation, we're looking at structured populations, association populations, and specifically today I'm going to talk about some results from looking at recurrent selection populations, populations that have been bred over time for specific traits. In general, we're interested in overall productivity and quality traits in the context of biofuels. The value of looking at these types of recurrent selection populations is that they're a complementary resource to discover genes and alleles that are important in the function of the traits that we're looking at. We expect in some of these open pollinated populations and land races to have maybe lower linkage disequilibrium than in other populations, so maybe get better resolution of the alleles. And we're interested in the possibility that there'd be rare alleles with relatively large effects that over a number of generations have been increased substantially in frequency. In addition, these are, I think, models for selection in natural populations because we have well-documented demographic histories and very clear selection criterion in these populations. The example I'm going to give today is an example related to seed weight. Seed weight is a core yield component in many grass species and in many other species as well. In general, as we looked at the increase in productivity over time, it's due to increases in plants per acre. But the overall yield is a function of the plants per unit area, the number of ears or seed pods produced per plant, the number of seeds per ear, and then as well seed weight. Seed weight has also been an important target of domestication in many species. Increasing seed size and organ size, for instance, have helped in the domestication. And the slide there, I have a picture of maize and teosinte, and you can see there's a dramatic difference in seed size between the progenitor teosinte and our modern cultivars of maize. The seed size selection program that I'm going to discuss was from a base population Krug. There's 30 cycles of divergent selection for seed size. I know with the microbial biologists in here, that might have been a one-day or a one-month experiment for you. This was a whole career of Bill Compton to carry out this experiment. So there's not a lot of these populations around, but I think they can provide some interesting insights. The selection method was phenotypic mass selection. I have some details there on the density of plants, relatively low density. Initially, the selection was based on visual observation of ears that had large and small seeds across the entire ear, so uniformity across the entire ear, and then later on in the cycles of selection, the selection was based on seed weight. Each generation there was 100 ears selected out of 12 to 1,500 plants, and in these populations the pollen came from all of the plants, and then the selection was based on the maternal seed weight, which will, I think, become important later as we look at the interpretations. As with many selection programs, the phenotypic response to selection was very impressive. I have some pictures of ears here. The small-seeded types are smaller than grains of popcorn. The middle ears that you see are the types of corn that you'd see on the trucks going by on the highway, similar to our modern hybrids that are grown. And the large-seeded types are quite large, similar to the corn nut snacks that you eat if you've seen those before. So there's a dramatic difference in the phenotype, almost a five-fold increase or 5-fold difference between the cycles of selection. In total, there's 60 cycles of selection since it was a divergent selection project for large and small seeds. We were particularly interested in this as well because in the large seeded population especially, the plants are much larger. Stalks are larger, leaves are larger, and so there seems to be a relationship between the selection for seed weight and for plant size. On this picture here, I have 2 ears, and those ears were produced by taking a mixture of pollen of the small seeded type and the large seeded type and putting that onto an ear of each type. So on the top, we have that pollen poured onto a small seeded ear, and on the bottom, we have that poured onto a large seeded ear. And on this picture, you can clearly see the effect of the pollen on seed size. There's a dramatic difference in the seeds that receive the large seeded and small seeded pollen on both the large and the small ear. In addition, even though there is this large effect, the largest seeds on the small seeded ear are still substantially smaller than the largest seeds on the large seeded ear. And that difference between the seed weights, there's various models, but is attributed to a maternal effect. And I'm not going to provide a lot of information to support this, but I think our observations are beginning to support that maybe 30 to 50% of the difference in seed weight that we see naturally occurring among these populations is due to the genotype of the mother plant. This might include imprinting, but probably other types of mechanisms as well. So we did an expression study on this population. We looked at various samples. We looked at seedlings because of the difference in plant size in the population. And then we looked at developing seeds 3, 6, 12, 21, 24 days after pollination. And the data I'll show you was based on a microarray that had a large number of genes of maize represented. Here's the clustering of that microarray study. In general, you can see that there wasn't a lot of difference between the 2 populations. At the seedling stage, they were quite similar in expression patterns 3 and 6 days after pollination, quite similar in expression patterns, and similarly 21 and 24 days after pollination. But at 12 days after pollination, there was a dramatic difference in expression pattern. I looked at the genes that were differentially expressed in those populations, and there are many genes that you might expect to be important, genes involved in starch synthesis and storage protein synthesis. Much of the maize kernel is made up of starch and protein. But as I examined it further, those genes were upregulated in the small seeded type relative to the large seeded type. And so in trying to understand this, we hypothesized that what's happening is that the rate of development is faster in the small seeded versus the large seeded type. And that rate of development probably is controlled again by the maternal genotype. We have some additional data now I'm not going to show today that's consistent with this hypothesis that one of the factors that's affecting the seed size is the rate of development in those plants. So then we tried to look at the differences in gene sequence and allele frequency using a whole genome paired analysis. And this is sequence that JGI provided for us. We gathered a large number of sequence, about over 100 billion base pairs for each of these populations. I have on the slide here the estimated, there we go, estimated depth of sequence that we're trying to develop. I'm going to make a few more comments on that, shooting for about 50x depth coverage in those pooled samples. I'm going to show you today just some of the results from the single end mapping, but we're also evaluating this with paired end mapping. With the first pass, we identified about 40 million SNPs in those populations, and the summary I'm giving today is summarized on about 15 million of those SNPs. So a very large diversity in these populations. When you look across various platforms of SNPs that have been characterized before, you see a large number, maybe at least 40 to 50% of the previously identified SNPs. So just in our simple calculation, we'd say the number of base pairs we generated, 100 billion base pairs, divide that by the size of the reference genome, 2.5 billion base pairs, we'd get somewhere around 43x coverage. But when we looked at the empirical distribution, it was substantially less than that. The median coverage was more in the area of 20 to 25x, depending on the copy number. And so what are the reasons that we might see this? And so here's some of the explanations that we have to get better, a closer match to that empirical information. When we look at the reference genome, this is something like this. The squares might be genes in the reference genome in between. Most genes in Maize are repetitive sequences. It's not to scale here. Probably only about 15 to 20% of the Maize genome is low copy sequence, with the remainder being highly repetitive. One of the things that's not completely accounted for in the Maize genome are tandem repeats, and that's illustrated by the purple lines on the slide. There's various tandem repeats that are present in hundreds of thousands of copies in the Maize genome. There's these knob repeats that account for a lot of the variation in genome size in the genome. In the reference, they're only accounted by one or a couple positions. And so when you take those into account, you probably have to add, I'm estimating here, just roughly maybe 15% of that overall genome. In addition, the pan-genome idea has been discussed in several talks already. Maize, there's certainly a pan-genome, a lot of genes that aren't represented in the reference genome. In a pilot study with just 21 inbred lines looking at the expressed pan genes, we identified hundreds of genes that were not present in the B73 genome, just in that small set of 21 lines. In another project with JGI here, we're looking more extensively across hundreds of inbred lines to use RNA-seq both to assist with genotyping and also to help to characterize the pan-genome of Maize. So if you put those together, the actual size that maybe should be in the denominator is much larger. I guess I made it much smaller, 3.2 gigabases. That would be a nice reduction in genome size here. And in addition, when you look at the number of bases that we characterized, about 15% of those map to other things than nuclear DNA, chloroplast, mitochondria, and so on. And so out of the initial calculation, we're getting about 50% X less coverage than we originally estimated. So that's one of the challenges of a genome like Maize. I think switchgrass is going to be very similar. In many of the outcrossing plant species, I think you're going to see this type of genomic variation based on the copy number of genes as well as repeats. And those calculations got us closer to the predicted, and there's some other factors in there that relate to mapping parameters and so on. I think that also helped to provide an explanation. So then how are we interpreting this allelic variation? What I've shown here on the left-hand side is a confidence interval curve to account for the drift that occurred during the selection process. So on the bottom, you have the initial allele frequency, and on the y-axis, you have the end cycle 30 allele frequency. If you have the same frequency in cycle 0 and cycle 30, you're going to be on the line. And to have a significant deviation from what you'd expect by drift, just now this is on a single locus basis, not corrected for multiple testing, you'd have to exceed these threshold markers here. There's a 95 and 99% threshold. As I said, the Krug population, and we're doing another population, golden glow is on the average of 100 to 200 females, 1,000 plus males, and so we're getting some respectable types of significance thresholds in those populations. Many of the other types of breeding populations have much smaller effective population size, and those boundaries are shown on the other side of the slide and probably aren't suitable for separating selection and drift. Just in terms of the depth of coverage, as I said, we're shooting for about 50x depth. These are 95% confidence intervals that you see with different amounts of coverage. In between 50 and 100x coverage, you're starting to really reduce those confidence intervals on your allele frequency. This picture is shown for an allele that actually has a frequency of 0.5. But you can see that the depth of sampling, the amount of information that you have, is very critical in estimating the allele frequency. So in spite of all those complexities and so on, what are the types of things we've observed? Here's just chromosome 1, and I'll show you some of the things that this exemplifies. It's consistent with many of the types of, with many of the other chromosomes. So one of the things, as I said at the beginning, that you might hope that you'd see from recurrent selection are alleles that would be at a very low frequency with a major effect, or maybe even a mutation that occurred, so it would have a frequency of 0 in cycle 0, and that would be fixed then at a high frequency through the selection process. We didn't see any examples of this in this program. It just wasn't enough generations, even though you see a lot of phenotypic divergence. It wasn't enough generations to see that type of a change in this material. For genes with more of an intermediate effect, you'd expect within a population selection program that genes of intermediate effect and intermediate allele frequency would be more likely to change, and so we're hoping to see things up in this area that would show a response to selection. And there's some candidates up there, but not a large number of things that showed a significant response to selection. And you can see that there's some targets that we're sorting through now in all these areas. As I said before, these significance thresholds were based on having a completely accurate sampling of the allele frequency at the end generations, and they expand a little bit farther when you take into account the depth of sampling. And so since each position has a different depth of sampling, these are just guidelines of what they'll have to exceed to be significant. But as I said, that sampling issue is quite critical at the end cycles. Just in contrast, in the previous slide, many of the alleles that we detected as SNPs had very low frequencies. We did require multiple reads with the same SNP to rule out sequencing errors or to minimize sequencing errors. If you compare that to a different platform where we looked at fixed SNPs that had been previously identified, you see a much different range of allele frequencies both in the initial and end cycles. And so I think this indicates that whole genome sequencing is allowing us to discover things that we weren't able to discover through other types of platforms that were currently available. Just a few candidate genes. There's many candidate genes you might consider. Some that are very well characterized in maize and other grasses are an invertase miniature 1 that reduces seed size as a qualitative mutation. The ADP glucose pyrophosphorylate subunits, brittle 2 and shrunken 2, have also been studied in many species in terms of directing starch into the storage organs. And so, and then there's multiple other genes in the starch pathway. And so here's a few examples of those. In these pictures now I'm showing the deviation of cycle 30 relative to cycle 0. There's spots going in both directions because this is based on the minor allele frequency. And as you can see, for many of these starch gene candidates, there's relatively little variation in these populations, maybe a little bit something for brittle 2 in the large-seeded population. But generally, these haven't been targets of selection in these populations. A few other starch genes, and again, consistent with them not being under selection. This was kind of interesting because these have been reported over time to be targets of domestication and have an impact in the increase in seed size, as we saw that maize went to teosinte. And that may actually be a reason that we're not seeing variation in these genes now, that maybe there's been a substantial reduction in variation over time. Some other types of target genes that looked a little bit interesting. FIE1, MES1 are genes that are imprinted in the endosperm of maize. It's kind of intriguing to think that maybe imprinted genes that are basically in a haploid condition in the endosperm might be more subject to selection. And another gene involved in epigenetics also shows some level of selection. This is a gene involved in the processing of 24 nucleotide small RNAs. These were intriguing leads when you look on a gene-by-gene basis, but I'm just going to show you one slide here that indicates in many parts of the genome, especially around the centromere, we still have large linkage blocks that are segregating in here. So even though we were thinking that these land races had substantially less LD than you'd see in other types of populations, and probably they do to some extent, in these areas of very limited recombination, especially in the centromere, you're seeing these pre-existing blocks of LD that I think are acting, that are present within these populations and affecting the selection program. So in some of those areas where I showed the genes that looked quite interesting, FIE1 for instance, there's either a selective sweep or a reduced recombination block there that you have a linked group of genes that we still have to sort through. So just some summaries then, some current interpretations of the pathways. One of the things I think we're finding with natural variation overall, and that this study exemplifies, is that some of the candidate genes that we think are going to be important are not the things that are showing up as underlying natural variation. And I think that's exciting because it provides then a mechanism to identify other genes and other things that are occurring in these pathways. As I mentioned with some of the starch and carbohydrate genes, and you might think of other classes of genes as well, things that have been targets of domestication may not be a substrate anymore for selection, as a lot of the variation has been reduced over time, and we might be looking at other mechanisms. As I mentioned, we're having various pieces of evidence that are indicating that there's a strong maternal influence on both seed weight and seed modifications. Much of the research in the past is focused on the seed itself. So here's a maize seed, the endosperm, the embryo, and so on. Within the maize seed, you have the pericarp, and then you also have the cob and all the other maternal tissues that this is attached to. And I think this is kind of the undiscovered territory now. It's not a well-characterized, these pathways are not well-characterized in terms of indicating how the maternal tissue is providing signals to the plant. To some extent, this makes a lot of sense. So here's the maize ear. It's been a very nice substrate for identifying single gene mutations. If you do an EMS experiment and self the ear, you can clearly see segregation on the ear. To identify the maternal factors that are affecting seed weight, you'd have to go another generation, self these seeds, compare among ears, and so it makes it much more complex, I think, to identify those genes using the types of screens that people have done before. So again, you're getting a complementary types of information from these studies of natural variation. The connection between seed and plant size, I think, is very interesting. There's numbers of genes where you have only a single copy, for instance, in maize, that has a function throughout the plant. So one of the ideas that we have with seed and plant size is that you have genes that maybe are involved in meristem size or organ size throughout the plant. In other cases, in other types of situations, we see the same type of thing where you have a single gene, for instance, in lignin that's responsible for lignification of a certain step throughout the entire plant. I think those genes are particularly interesting targets because they might be a place where we can modify expression, so we have one type of expression in one part of the plant, another type of expression in another part of the plant. And so these are the types of things that we're also finding through these studies of natural variation. And then just the final summary, and which I think links in good with the K-base. You have all this information, it's very hard to interpret it, but I think there's lots of valuable insights that you have when you put these data sets together in complementary ways. And the data sets that we had here were both phenotypic data as well as genetic and genomic information. And we had to look at that in multiple contexts and multiple ways to begin to get the interpretations that we're making. And I think that's the type of thing that these databases can really support to help pull together those diverse phenotypic and genetic data sets. So that's all I have. Thank you for listening to me.
I think we have time for one question.
It seems like it might be, I don't know if you've done this already, it seems like it might be useful to gather some more phenotypic information about the kernels, like the nutrition facts, which could be modeled against some of these genes as well, so you understand if the size.
Yeah, so one thing that's interesting in these, we've looked at starch and oil and protein, and the concentrations are similar in the large-seeded and the small-seeded type. And so in this case, it seems like it's primarily a scaling issue where the entire seed has just become smaller. We're also looking across different generations to try and also better separate out the maternal and endosperm express factors in various ways also, and then also looking at the components of those. Yeah.
So just a quick question. Did you look at whether or not the differences in size were due to increases in cell division or expansion or perhaps endoreduplication, like what Bob was speaking about today?
I have not yet, but we're very interested in looking at that.